Okay, thank you so much for the invitation for the presentation. Very nice words. Thank you to Professor Visca. I talk today only of one bacteria species. The name is difficult to pronounce in English. There is a debate if it's Klebsiella pneumoniae or pneumoniae. If one knows the Latin probably is pneumonia. And I like this material because this bacterium because it is occupying mostly the last 3 years of my daily work. And since I am in Sapienza, it's really now fascinating me. So I try to introduce you to this antimicrobial resistance mechanisms and the evolution of the Klebsiella pneumoniae that is reaching the pan resistance, means resistance to all antimicrobial classes. It belongs to this Klebsiella pneumoniae species complex. Here it is. And this is a phylogeny built by the group of Dr. Holt in Australia. She is doing nice work for the phylogeny of this strain. And you see is an Enterobacterialis, and there are other species that have impact on human health, but surely the most relevant is Klebsiella pneumoniae. And if you look at the plate here, a piece of plate, this is a McConkey plate. It means that you see the color of the lactose fermentation. But what you should note is that very often this bacterium has very wet mucus colonies. And this is really a peculiarity of the Klebsiella pneumoniae of clinical origin. They are mucus because they produce a capsule. And I remind you that the capsule in gram eggs is made by a polymerization of saccharides, then is exported by an ABAC system outside and linked with a lipid to the outer membrane. And this is synthesized in Klebsiella by clusters that are normally encoding for a constant part and the variable part. And the variable part is particularly interesting because it makes different assembly of different sugars. And depending by these genes, you can have different capsule outside the outer membrane and different key types. So it's used to typing Klebsiella by Witzy typing. This was set up by the Institute Pasteur by Professor Brisse, and it consists simply in sequencing some genes of the capsular clusters and to determine the alleles, but also it's classified by key types. These methods are useful to understand if there are phylogenetical linked groups and with easy methods. But the most frequently used is the multi-level sequence typing. I will talk to you in a while. We divided Klebsiella pneumoniae in two major groups. One is the hypervirulent, in which the capsule is overexpressed. And this is the list of hypervirulent associated genes. Most are clusters encoding for iron uptake systems. And then there are two regulators that are overexpressing the capsular cluster, and these are normally located on a virulence plasmid. This is the aspect of an hypervirulent, hyperviscous Klebsiella. You cannot touch the colony with a loop. You get a streaming test positive. And this is particularly indicative of how can be aggressive this Klebsiella. In fact, The hypervirulent Klebsiella can cause infection also in healthy human beings. This virulence is different from the normal strains that we study in the hospitals, called classic Klebsiella. The classic Klebsiella are not so virulent. They can be capsule positive, but not hyper producers. And they can be normally colonizers of the gut of patients during their stay in the hospitals. But continuing hospitalization, and when patients are really sick and with some immunosuppression, these kind of strains can cause serious infections, mostly urinary and pulmonary infection, but very often also sepsis. This kind of Klebsiella is defined opportunistic because it is not a frank pathogen, but can cause serious diseases to patients with, that are fragile with other disabilities. This is an important point of reflection because the differences I just described you can be seen also considering all the major clones that are circulating in the world. And Klebsiella pneumoniae has been identified in two groups, also by genomics. You see that there are some core genes that are common to all Klebsiellas, and there are some virulence, some strains that are positive for virulence genes, while others, the classical Klebsiella pneumoniae, are not so virulent, but they are hungry of antibiotic-resistant genes. And they can collect up to 12, 13 different genes conferring resistance to mostly all classes of antimicrobials. We will talk mostly of this kind of Klebsiella. And these clones have been defined high-risk clones because they are a global problem. They are not only Italian or European. There are some clones that were widely distributed on the planet. And they are classified by multi-locus sequence type in clonal complex clonal groups. And you see here some numbers that I will use to classify them. And we will talk of the clonal group 258, clonal group 307, clonal group 147 mostly today. This is the definition of high-risk clones, those that are distributed along the world. And you see this is Italy. We experienced some outbreaks of high-risk clone Klebsiella starting from 2008. And different species and different types have been collected during the years. In this report, you see mostly we had 258 and 307, as I told you. Okay, so this was my brief introduction of the bacterium, but now I will continue to talk about it, but through my vision, which is the description of how some kind of bacteria like this one can acquire resistance to antibiotics. And I would like to start with beta-lactams because beta-lactams are a wonderful class of antibiotics. You know, beta-lactams are normally inhibits the so-called penicillin-binding proteins that nothing else are that the transpeptidase that catalyze the della transpeptidation. And the link of these antibiotics with this protein makes this link impossible, and therefore the bacteria are fragile because the cell wall is not well built. And this is due to the fact that beta-lactam structurally reminds the della structure, and therefore they can substitute della into the pocket, the functional pocket of the transpeptidase. Starting with penicillin, discovered by Fleming, there was a race between the human beings producing antibiotics and bacteria finding resistant to those antibiotics. And especially for the, oh, sorry, it's too much. Okay. Especially for beta-lactams, we have a list of new generation beta-lactams along the years that have been produced by pharma, introduced into the market and used. And for instance, we had in the 70s, we had the second generation cephalosporins, and then third generation cephalosporins, fourth generation cephalosporins, and fifth generation cephalosporins in recent years. Then we had a completely new structure of beta-lactams when carbapenems were synthesized. And we had imipenem, then we had meropenem and ertapenem along the years. And recently, we have the new generation beta-lactams combinations in which previously used carbapenems or third generation cephalosporins are combined with inhibitors. Inhibitors of what? Inhibitors of the mechanism of resistance that give resistance to all the previous beta-lactams. This is the situation we had before 2008. We already had isolates. This is a map of the European SCDC, which survey every year the situation of resistant bugs, especially in invasive infections. This is a map of 2006 for Klebsiella pneumoniae with resistant to third generation cephalosporins. This map clearly shows you that we reached very soon in the food resistant to this kind of drug. And in the same period, we had full susceptibility to carbapenems. And therefore, carbapenems were really highly used to cure Klebsiella pneumoniae infections showing third generation cephalosporin resistance. The use of carbapenems started in these years. Immediately, it was observed a slight but constant development of resistance to carbapenems as well. We observed in 2009 an unusual number of positive isolates of this kind of clone, which had a differential resistance to carbapenems. I mean, they were not resistant to whole carbapenems. They were resistant just to one of those, the ertapenem, but they were susceptible to imipenem and meropenem. I was curious to study these strains, and this was a nice work in which, with the technology of the time, we identified some a mutation into the loop tree of the outer membrane protein. And this is the porin that allows the diffusion of the entrance of the beta-lactams in gram-nex. It's an important protein that is in the outer membrane. And this was the duplication of 2 amino acids in one of the most conserved loop comparing sequencing of all bacteria. And we, at the time, we didn't understand exactly the role of this duplication. But then, now, for instance, mostly all strains have a defect like this in the porin. Why? Because this is the highlight of the porin, and it was with a restricted lumen. Therefore, ertapenem, which was the largest molecules, physically largest molecules, was filtered out, while imipenem and meropenem enter into the cell. This was the first observation of a partial resistance to carbapenems. And then the other mutants were observed, in which the porin was completely eliminated by a premature stop codon, and these were going on ertapenem, imipenem, meropenem all the carbapenems in a while. This situation completely changed in around 2011 because we didn't observe anymore the porin mutations, but we observed the entrance in Italy, and I was surveying the hospital in Roma with the collaboration of some societies that gave me the strains. We observed the evolution to full highly level resistant Klebsiella pneumoniae to carbapenems, thanks to the acquisition of a carbapenemase. And the carbapenemase is KPC, because it's a Klebsiella pneumoniae carbapenemase. It was discovered for the first time in the United States, in hospitals in New York. Then there was a big problem also in Israel, and then also in other countries. This is the map dated 2016 of the country that were affected by KPC producing Klebsiella. And you see, we were among those considered endemic for the spread of KPC, as well as the United States and South America and East countries. And this is the situation of 2017, and changing completely the prevalence of carbapenem resistance in Italy, and bringing it to a range of 40% in all the hospitals. So as you see in this map, we had a big problem of infections sustained by this kind of Klebsiella producing KPC, and then being resistant to all beta-lactams. And this is a study that was published in 2019, but refers to data of those periods, so in 2015. And it sees, you see, this map is another map. This is the map of the so-called dailies. Dailies are the disability adjusted life years. It's like a measure of the true years lost because of antibiotic resistant bacterial infections. It means that this is a net to the previous condition of the patient, and the relevance of these data were pushed Italy to take measures, and this is the reason why we have now a plan for the control of antimicrobial resistance in our country. You see, we were the first bar in this paper with 2,000 cases and 10,000 deaths. And if you look at which colors are filling this bar, the pink is third generation cephalosporin E. coli, Klebsiella, and Acinetobacter pseudomonas. And the green is carbapenem resistant, now Colistin resistant E. coli, Klebsiella, and Acinetobacter pseudomonas. These 4 gram-negates are responsible for almost all the problems in our countries. Of course, I will talk only of Klebsiella, whose impact is this one. And this is the effect of gram-pos. Therefore, there is a big problem. There was a big problem in our country, and the reason why it was so carefully studied. In this study, there was an important surveillance study in which strains were conferred to ACDC. So we have a good point of the situation in 2013-2014 in Europe, in which Klebsiella pneumoniae were collected by 32 countries, 244 hospitals, 1,700 strains. Italy provided just one type. It means that we were really under an outbreak due only to KPC producing strains. The other countries, you see, had more variable situations with different carbapenemases, with different mechanisms of resistance. We had practically one clone. In fact, in this paper, they compared the clone circulating in Italy, which is blue, with those identified in Israel and United States. We were experiencing the epidemic circulation of clonal group 258, composed by mostly two strains, 258, and the highly relative 512. We contributed by completing the sequence of the plasmids carrying KPC. The plasmid was more than 110 kilobases, and the KPC gene was here in a transposon. The plasmid was an INCAF plasmid with two replicants and the classic mercuric locus, and no other relevant resistant genes on it, except the carbapenemase. But within these cells, there were other plasmids. One very large was 208 kilobases and contributed to resistance of the clone, bringing additional resistant genes, conferring resistance to all drugs or not used drugs, but also to copper and silver. This pattern, I thought, this comes from environment, this comes from other sources that are not clinical or nosocomial. And there was also a virulence factor, an iron uptake system here on the plasmid. There was a third plasmid contributing to aminoglycoside resistance that was unstable. This was also a problem because sometimes the strains were treatable with aminoglycoside, sometimes not, and it was not clear. But looking at the small plasmid that can be unstable, you have, we clarify why gentamicin was variable in this clone. Suddenly, in 2016, Caterina Mammina from the hospital of Palermo phoned me and said, Alessandra, something is changing. We were annoying to report 258, but now something is changing. We saw many 307, which is another clone. And can you investigate if other hospitals are looking or are seeing this 307? So I did more. I called the University, the Institute Pasteur, Sylvain Brice, and the HPA in London, and also I read some reports from Colombia, and we all together established that yes, there was a new KPC producing Klebsiella pneumoniae that was starting its spread and changing the epidemiology of 258 with 307. And simultaneously, we studied this clone, and you see another paper in Texas, in Houston, was declaring an unexpected abundance of clonal group. Why I report this? Because it's a nice evolution when the strains, the clones change themselves and in the epidemiology. And we analyzed this clone. It was bringing Yersinia bactin. This siderophore was an integrative conjugative element with a type 4 secretion system integrated in the chromosome. There were two types of S307 with two different integration into different loci in the genome. And we studied the plasmids of 307, and the same identical plasmid carrying KPC with the transposon and the mercury ions was identified in the clone, while the plasmid carrying inside together with colocalized in the same cell with this was different, and it has much more resistant genes, including the extended spectrum beta-lattamase resistance, CTX-M-15, and also 4 new virulence factors, including glutathione importation, iron uptake, urea transporter. I remember you that ureases are in the chromosome, and these are just transporters specific for urea, and therefore this strain had a sort of commitment to urinary tract infections than the other, and also a rare cluster for the synthesis of glycogen, which was located on this plasmid. And the situation of the 307 producing the standard spectrum beta-Lattamase CTX-M-15 was described also in other countries in the world in the years immediately after our first indication. What happened? Simply, the 307, which was underscored because not carbapenem resistant, but just producing a standard spectrum beta-Lattamase, had the transfer of the plasmid carrying KPC from 258 clone. This transfer made probably this clone more successful than the previous one, and it emerged in the group of carbapenem producers that are normally under surveillance. And in a very recent paper in 2021 are arriving national data from the 2018-2019 years. And you see now 307 is in all the country almost reaching 20%. Still 258 has been reported, but other clones are reaching the stage acquiring KPC by bacterial sex, by conjugation. What can we do then when beta-lactams don't work? There are a list of other drugs that can be used. At the beginning, the most popular was Tigecycline, now is Colistin and other polymyxins and some aminoglycosides in a minor way, and Fosfomycin, and then also the new drugs that just entered into the market in 2018 in Italy, ceftazidime, avibactam, and meropenem. I will tell you something about Tigecycline, Colistin, and Fosfomycin resistance in this strain. Tigecycline is a nice drug, is a derivative of tetracycline with the addition of one group. This group has been included in the molecule because it makes two links with the 16S RNA instead of one done by tetracycline. And this tight link binding inhibits the protein synthesis and the cell die, guys. This is the model of the ribosome, and this is the 16S, that is like the ham in the sandwich, in the middle of the two subunits. And this is the imaging of the Tigecycline linked to the 16S. We were among really one of the few describing the Tigecycline resistance in Klebsiella pneumoniae ST512, so the same with current KPC. And the reason is that using Tigecycline against the KPC producers, suddenly we observed this few, but very first resistant strain to this drug. And I had the idea to draw to Pfizer, which was the producer of Tigecycline, asking money, to say, I have the first pairs of strains, one susceptible, one resistant, isolated from the same patient during treatment. Are you interested to see them by genomics? And they said yes. And so in 2014, when we did genomics of Tigecycline resistant strains, and we found two mechanisms of resistance to this drug in different pairs of being sequenced. So there were different mechanisms. One was the inactivation by premature translation by a stop codon mostly in RAM-R, which is the negative regulator of RAM-A, which is the positive regulator of the SCR-AB efflux pump. The efflux pump is here, the drawing is the way to decrease the intracellular concentration of the drug efflux out actively, and is done by two subunits that are encoded by this cluster. This cluster normally is not expressed except when RAM-A is in a condition of activation, and therefore with the RAM-R RAMR depletion, all the efflux pump was actively produced, overproduced. The other mechanism was in a couple of strains that we couldn't find any problem to efflux. And therefore, the unique mutation we had, comparing the susceptible and resistant strain coming from the same patient a few days of distance, we found this mutation just binding to leucine substitution in a small protein, ribosomal protein S10. If you look again to the model, S10 is this protein. I was disappointed to see that the protein was so far, so lateral in the position. But then looking better, it has a long head, and this is the leucine, which is mutated. And in fact, it was exactly the first amino acid less than 10 angstrom to the site where Tigecycline should bind the 16S. And this mutation perturbed the link, and the strain was susceptible. This was the first demonstration of site mutation that caused this resistance to Tigecycline in a protein and not in the 16S itself, by itself. Colistin. Colistin is a nice drug that is able to bind and the LPS subunits and disaggregates and destroy the outer membrane first, and then the inner membrane. And also neutralize the LPS toxicity because of these subunits are neutralize the toxicity. The mechanism of resistance is very complex because this drug is like a detergent. It acts outside the cell. It doesn't enter the cell. So there is no problem for the entrance, but the mechanism of resistance is very sophisticated. And it consists in a decoration by arabinose or phosphate-enolamine of the LPS. The decorated LPS is resistant then and can be binded by colistin. It has been discovered also a plasmid phosphate-enolamine that can confer high level of resistance to colistin. I pass to Fosfomycin because the resistance mechanism will be described in one strain that I recently studied that is impressive. Fosfomycin is another nice drug. This is a structure is imported by two transporters and normally acts as a mimic of glycerol 3-phosphate or glucose 6-phosphate, again in the peptidoglycan synthesis, and is a substrate for muA. Thus, it blocks the construction of the cell wall and the cell die. The mechanism of resistance has been described with different systems, again transporters, or by the acquisition of a gene which is able to deactivate, degrades the fosfomycin drug. And I now give you really the dimension of some problems at the hospitals, like it happened in Tuscany in 2018 and 19. There was an enormous outbreak in several hospitals of this region due to another clone, which is ST512, producing the Metallo Beta-Lattamase NDM. You had probably in those years the problem of Tuscany outbreak, and this prolonged outbreak caused several deaths. And the NDM-1 beta-lactamase is a carbapenemase, highly potent, is not inhibited by any inhibitor, and just the unique drug that works is azeturnem in association with other drugs. And these strains were susceptible to colistin, tigecycline, and Fosfomycin. In 2020, I was involved in the typing of a new clone of ST147, which was isolated in Tuscany. The difference with the previous Outbreak clones was that, yes, it was still NDM producers, but it had acquired resistance to Colistin, Tigecycline, Fosfomycin. And it derived, these are the two strains isolated in 2020, and this is the phylogenetic tree of the of some strains of the Outbreak. These have been kindly offered to me for this comparison by Professor Solini from Firenze, from Florence. And you see the new clone derived directly from the previous one, and there were very few SNPs that discriminated the new isolate with the old isolates. And in these strains, we found a depletion of the MGRB, which is the regulator of the arabinose cluster. And this depletion means hyperactivation of decoration and colistin resistance. We found the RAM-R depletion, which means, as I explained to you before, overexpression of RAM-A, and RAM-A means overexpression of the efflux pump, ACRB, and Tigecycline resistance. And we found also the mutation into the GLPT transporter for Fosfomycin that consequently mean the Fosfomycin resistance. With 4 mutations, the previous fully resistant with the Carbapenemase NDM strain evolved and acquired 3 drugs that were used against this strain. Conclusion. In this timeline, I described in this review that we wrote with my friend Shiri. Shiri Navomenatia is the person in Israel that along these 20 years worked on the same strains I did, the ST512, ST2058. So we are friends since some time. We collaborate on the PK, PQIL, plasmids, KPC, and so on. And we decided in 2017 to write a review on Klebsiella. And just reading papers, we reconstructed this timeline in which the introduction of drugs and the discovery by Klebsiella of resistance was, as you see here, timely. I mean, as soon as first generation cephalosporins were introduced, Klebsiella found chromosomal mutations. But then she got the first beta-Lattamases, and these were even more efficient when second generation cephalosporins arrived, and then the third generation cephalosporins, and then the fourth generation cephalosporins, and then the carbapenems and the carbapenemases. And these were the genes she obtained from the pool of resistant genes and evolved in resistance. This is just the panel for beta-lactams. What we did for all the other enzymes, more or less, is the same. What is interesting to see is that the genes arrived with the plasmid, but then they moved to the chromosome. Very recent genomic studies demonstrate that Klebsiella is going into this evolution of integration of acquired resistant genes and a lot of mutations that confer resistance. And this is due to the fact that it spreads in clones. So the horizontal transfer, which is highly relevant for other species like E. coli or other species in gram-nex, in Klebsiella, since the spread of the successful clone is so evident, these clones with the time evolve in the integration into the chromosomes of the minimal needing for resistance. This is therefore the evolution of this Klebsiella, starting with efflux pumps modifications and altered targets sometimes. It evolved to XDR by acquisition of plasmids. And these plasmids are not just for itself, but they can be transferred to other bacteria. They can be transferred to between, there is an interplay between strains. And so some strains prevailed with the particularly intention and selection with their own carbapenemase. KPC was characteristic of 258, NDM of ST147, and OXA48 for another clone that I didn't talk today is ST15, because we didn't have so many infections by the ST15. And now we have Klebsiella sometimes with these carbapenemase genes, like NDM, for instance, in companion animals. We had dogs described in Switzerland that are probably receiving these clones from the owners, but it's not so clear because we have also some identification of carbapenemase producers in food, in other kind of animals, and in the water, in the sewage, and of course in the hospitals. They are very serious problem. We are now observing more strains because patients are screened for colonization. Because the first step for Klebsiella is to colonize the patient silently, especially those in long-term care facilities and nursery homes. And when the patient has a serious problem or it goes sicker, they can cause serious infections. And I close with this imaging of Klebsiella, which was the winner in 2016 of the American Society of Microbiology Agar Heart Contest. It is, the colonies are these of the cherry tree and the flamingo body. And these are my group. This is my group currently. These are the colleagues. Most of the work has been done at the Istituto Superiore di Sanità, but more recent work is now I'm doing in Sapienza. I am in this villino, is a villa in San Lorenzo, very close to Sapienza, but not inside the university campus. I am working with professionals. This is a medical doctor, and these are the dirigent biologists working at the hospital. They are 4 students of at the last year of the School of Specialization in Microbiology. And this is my student of science. She is studying biotechnology. And we have now a sequencer, a nanopore sequencing running in the villa, and we are doing a lot of things with these younger guys. I'm so happy to have restarted my second life in Sapienza, and I would like to thank a lot of persons, of course, Guido Antonelli for he hosted me, and Professor Bianca Colonna, because she's very helpful for daily life in my new experience as a teacher, as a professor, and all of you for your patience. Thank you.